So here we are, Eric,
Tim.
Christine.
and we're all here, and we're going to give you a short review on the paper, Enzymatic Amplification of Beta-Globin Genomic Sequences and Restriction Site Analysis for the Diagnosis of Sickle Cell Anemia. For our introduction, we're going to talk about why the study was performed, what they needed to have already in place to make these changes to the scientific knowledge, and why this study was important. As in all scientific studies, the researchers came into this one having a significant amount of prior knowledge and prior techniques that were built on the work of other scientists in this field. Firstly, the researchers had a firm understanding of the central dogma of biochemistry. They understood that DNA went through a self-replication process involving polymerase, free nucleotides, and priming sequences. They also understood the process of transcription, by which DNA coded for RNA, as well as translation, in which RNA is coded for protein. And they also understood the functional aspect of protein as a fundamental building block of life, particularly in this case, how protein makes up hemoglobin and red blood cells, and how mutations in DNA can result in changes in protein structure. The researchers used several techniques in the lab to help them get their results. One of these techniques is the Southern blot, which combines DNA fragmentation, gel electrophoresis, and hybridization to analyze specific DNA sequences. The resulting product of the Southern blot is labeled bands on a piece of paper. The researchers also understood and had available to them restriction enzymes that could cut DNA into fragments depending on the type of sequence that these enzymes were specific to. For instance, they could use D, D, E, and HINF to digest at sites such as CTCAG and sites GACTC. This was essential to their research. Finally, the researchers could use oligomer probes to tag sequences of DNA with fluorescent markers that could be used to resolve the Southern blots such that they could visualize at our scale what was going on with DNA. In this graphic, there's a specific marketed light cycler probe that resolves when you irradiate it with some form of specific light wavelength, and you're able to see the probe targeted on the DNA. This use of labeled oligomer probes was done in this paper as well when they used the phosphorylated probes to note the presence of a specific sequence of DNA. In this study, researchers were specifically looking at whether sickle cell disease could be diagnosed using this novel laboratory technique that involved the analysis of DNA sequences. To understand that this was possible or feasible required an understanding of the sickle cell disease process. As we mentioned before, the researchers understood that DNA coded for RNA coded for protein, and understood that changes in DNA or mutation therein would result in changes to the protein structure. They understood that sickle cell disease, which was named for the sickle shape that they could visualize using light microscopy of red blood cells in human circulation, was a molecular disease that resulted from DNA changes. Here you can see the red blood cells of a healthy individual without sickle cell, hypothetically healthy, And here you can see a graphic or a cartoon that's representing sickle cell disease. The sickle cells tend to aggregate together, stick to the walls of the vasculature, lyse and leave toxic byproducts, and all in all result in an unhealthy individual who has circulation issues. Here we can see a graphic that's representing the mutation that occurs in sickle cell disease. You can see here the change in the normal and sickle trait with a mutation at the alanine to thymine, changing the codon, resulting in a different amino acid being placed in the final hemoglobin protein. This change in the protein structure results in a misfolding or a change in folding. The change in the folding results in these sticky sickle cell hemoglobins that you could see on the prior graphic, and these aggregate and form the disease. We'll talk a little bit about why this matters. Why is this research important? And this paper in particular was chosen as our project because it was pivotal in developing some modern cellular techniques. The most important aspect of this paper involved the utilization of PCR or polymerase chain reaction to a useful lab technique in the diagnosis of a disease. Polymerase chain reaction involves the denaturation, annealing, and extension of DNA strands repeatedly to increase the physical number of these strands such that they are easier to resolve at our scale.
Methods and techniques. Two procedures were used for the detection of sickle cell mutation. The first procedure involved amplifying specific beta-globin DNA sequences with the use of oligonucleotide primers and a DNA polymerase. This is also known as the polymerase chain reaction procedure, developed by Melissa and Faluna. And the second is the solution hybridization of the amplified DNA with a specific oligonucleotide probe and subsequent digestion with a restriction endonuclease. The first procedure involved a primer-mediated enzymatic amplification of specific beta-globin target sequences in the genomic DNA, which resulted in exponential increase of target DNA copies. In this procedure, 11 DNA samples were derived from lymphoblastoid cells or white blood cells that were analyzed for beta-globin genotype. They were identified as AA, which was the beta-A wild-type beta-globin allele, the SS, the beta-S homozygous sickle cell allele, and the AS, the heterozygous sickle cell allele. A southern blot was prepared to show the degree of specific gene amplification that indicated an overall efficiency of approximately 85%. The second procedure was the oligomer restriction, which is a procedure used to detect an altered DNA sequence or allelic variants in a genome. For the diagnosis of sickle cell anemia, a probe was designed to be complementary to a region of the beta-globin gene locus surrounding the 6 codon, which is where the beta-S, the sickle cell allele, and the beta-A, the wild-type allele, differ. DD1 and HINF1 restriction enzymes were used to generate specific cleavage products of each allele. By digestion with the DD1 restriction enzyme in the beta-globin allele AA, a labeled 8-nucleotide octamer will be produced. This short 8-nucleotide cleavage product will then dissociate from the genomic target DNA. In the SS homozygous sickle cell allele, the DD1 does not cleave due to the base pair mismatch, and the HINF1 digestion will cleave at the site to release a labeled 3-nucleotide trimer. In the AS heterozygous sickle cell allele, both a trimer and an octamer will be detected.
Now that we've covered what was known prior to the experiment and the methods that were used, we can dive into the results. So figure 1A and B are depictions of the probes, primers, and beta-globin target sequence along with the restriction enzyme sites. RSO6 is complementary to the wild-type sequence of beta-globin, but not to the flanking PCR primers PCO3 and PCO4. PCO3 3 is a primer complementary to the negative strand, and PCO4 is a primer complementary to the positive strand. And you can see the two primers here, and then you can also see them here along the beta globin target sequence. There's PCO3 and there's PCO4. There's another probe, RS10, which was referred to as the blocking oligomer, and is added to bind to excess RSO6 to prevent non-specific cleavage of potential binding to non-target DNA. RSO6 and RS10 are similar except for the 3 nucleotides that are depicted with the down arrow here. In figure 1b, you have the HINF1 and DD1 restriction enzymes, where HINF1 is an invariant version of a restriction enzyme, and DDEI is polymorphic. Figure 2a and b are a southern analysis of PCR-amplified genomic DNA with the RSO6 probe. So only the lanes here with at least one wild-type globin gene or allele show a distinct mark at the 114 base pair marker indicated by the horizontal line. Lanes 3 and 4 do not show because they do not have any copies of the wild-type globin genes. So lane 1 is MULT4, which is homozygous for wild-type. SCO1 is the heterozygous copy. GM2064 is the null, so it has neither the A or S allele, and then you have unamplified DNA in lane 4, which again will show nothing because it's not amplified. It's not strong enough of a signal. In figure 2b, what they are trying to do is test the efficiency of 20 cycles of PCR amplification. So using differing amounts of beta globin sequence, they used it to represent each of the different efficiencies ranging from 70 to 100%. So lanes 2 to 8 are efficiencies ranging from 70 to 100%, and they use that. They compared these results to a standard. After hybridizing with the RSO6 probe, the overall efficiency was 85%, which you can see these two lanes are very similar in strength, and that's kind of how they came to that conclusion. But lane 1 serves as a control because it's homozygous. It uses the MULT4 cell line, and that serves as a basis for determining PCR efficiency. So again, lane 1, lane 5 have similar signal strengths, and that is how they came up with the 85%. But how do they determine the 85%, or what does the 85% mean? So that means, so if there was 100% exact copying of each amplification, then you would end up with roughly 1 million nucleotides matched. With 85% efficiency, you get roughly 220,000 matching nucleotides. But for this experiment, that was really good. So in figure 3, the goal of these figures was to show how specific probe cleavage products for each allele were generated using oligomer restriction. So figure 3a shows beta A contains a DDI restriction site which acts as an endonuclease and creates an 8 base pair fragment exclusive to the beta A allele, and then figure 3b shows the same thing except for the beta S. That one is digested though with HINF1 restriction site and acts as an endonuclease and creates a 3 base pair. If you were to have a heterozygous version of these two, then you would get both cleavage products. So you would have an 8 nucleotide cut and then a 3 nucleotide cut. So figure 4 demonstrates the same thing as figure 3, except they used a different method using sequential digestion with cloned beta globin genes. So again, you have the homozygous wild type here, heterozygous, and then you have homozygous mutant. So this figure A and B illustrates how the beta globin genotype is identified in human DNA with the PCR and oligomer restriction method. So lane 1 depicts an unamplified MULT4 DNA, which MULT4, the cell line, is homozygous wild type, but since it's not amplified, you're not going to see a signal, and that's why it was used as the control. Lane 2 is the amplified version of MULT4, and again is the homozygous wild type, so that's why you get the 8 nucleotide signal sequence here. Lane 3 is the SC01, which is homozygous mutant, and that's why you see the 3 nucleotide signal. And then lane 4 is the GM2064 cell line, which is null, meaning that it doesn't have either the A or S allele, and that's why you get no sequence. Lanes 5 through 11 depict the same things that are shown here in 1 through 4. They just use different cell lines just to confirm that they got appropriate results. So anywhere where you see just an 8 nucleotide signal sequence, that means you have homozygous wild type. If you have both the 8 and 3 nucleotide sequence or signal, then you have heterozygous, you have the A and the S allele, and then if you only see a 3 nucleotide cell signal, then you have homozygous mutant. Figure 6 just shows how important it is to have many cycles of amplification and how the effective cell cycle number is on cell signal strength. So at 15 cycles, you obviously can hardly tell that there are binding patterns here in like 1 and 2, and then also in 3. But then you can very clearly see at 20 cycles the different nucleotide signal sequences. So this sample was CH2 and was homozygous for the wild type. The sample 12, which was heterozygous, shows both the 8 and the 3 nucleotide as expected, and then sample 5 has the 3 nucleotide sequence, which is homozygous mutant. So it's just again showing how the more cycles you have, the greater the signal strength you have. Lastly, figure 7 depicts the varying amounts of genomic DNA of a heterozygous individual as a template for PCR amplification. So they were doing amplifications of DNA with large amounts of DNA, and they wanted to see how little they actually needed to still see a signal. And what they determined is that they needed 20 nanograms or less to detect the presence of mutant forms of the beta globin gene, as shown in lane 3. And then as in lane 5, they just use this as a control because GM2064 again is null for both the A and S allele, so you wouldn't see anything. But yeah, just as little as 20 nanograms is needed to detect the mutant alleles of sickle cell anemia. Next, Natty will do the discussion.
Hi, this is Nathaniel, and I am doing the discussion part about this article. So, where the hypothesis supported? The hypothesis is the prediction they make before the experiment, and in this paper, there is no specific hypothesis was defined. What did the author conclude? So they conclude that they could use PCR within DNA to create a measurable signal that is useful for the diagnostic of sickle cell disease within a small sample of genetic material. Uh, they noticed that restriction enzymes could be used to effectively determine differences in DNA, and they used these methods to effectively test for sickle cell coding DNA, which will lead to the disease. And I believe they also conclude that this technology may be useful in diagnosis of the other diseases. Are there any drawbacks to the current methods? Of course, there were some negatives to the current methods. And first, the current method required prefabricated restriction enzyme of the known function. And second, there are known issues with the oligonucleotide in terms of specificity. This ties into the lack of mutation erasions of their testing. And third, there are moral questions of whether having a genetic information about the individual might lead to discrimination against those with disease. And finally, I believe while these methods of testing is speedy and accurate, it's limited due to the requirements for a specific laboratory environment, and it's not speedy or quick. What is your opinion of the study? Is it sound? What did you like, dislike? We agree that this study is good. We like the study presented the information and is completed with the topic, and they put a lot of detail in every single step, and they put some figures to make us understand what they're saying. And like I said, they went really deep, and it's like what 6 pages article. It has a lot of information, and they give us an example of some of the topics what they're saying, so that will make us understand a lot better, including the figures. And the negative part about this article is just like I said, they didn't provide any hypothesis, which we which might not quite good thing, because that's like the main part of the article should be, because like they think about this is going to happen, but it might turn out happen or it might not turn out happen. So it's just a lack of hypothesis. But other than that, it's a very good article. How are the findings and conclusion relevant from the practical perspective? Relevant in that they provide proof of the concept from PCR, on which other genetic testing are based, and also because they provide a method of diagnosing sickle cell disease from a small sample of genetic material. In principle, other genetic diseases may be diagnosed using a similar technologies. Do they support or refute an exciting theory? Their testing supports the genetic theory of replication of DNA, the use of polymerase adding nucleotide into a polymer. This theory is supported because it's a fundamental to their results. How does the work contribute to our current body of knowledge? What should the future research focus on? Definitely, this article, this paper should tell us more about our body knowledge. Should teach us a lot about our body knowledge, and in the future studies might include a series of preclinical studies to establish whether a viral vector can correct a defect in blood cells for patients with sickle cell disease.